Okay, so let's talk about some non-membranous organelles. So the very first one is ribosomes. So ribosomes are considered the protein factories, and they're made of RNA, specifically the rRNA, which is ribosomal RNA, and the proteins. So they are involved in the production of the protein. So that is why they're called the protein factories, and these are the non-membranous organelles. Moving next, we have microtubules, microfilaments, intermediate filaments. These are all protein filaments, and they make the skeleton of the cell. So the way we have the skeleton, which is made up of bones and the muscles, similarly every cell has a skeleton, and that's called the cytoskeleton, and that is made of microtubules, microfilaments, and the intermediate filaments. Okay, moving next is your okay, let me go into the slideshow mode. So these are basically your, you can see here, this is all the cytoskeleton. So this is made of your intermediate filaments, microtubules, they all together make the skeleton of the cell. Now next is what are your centrioles. Now these centrioles, like around the nucleus, like outside the nucleus, there's a region called centrosome region, and this centrosome region has these two centrioles, and they are perpendicular to each other. Now these centrioles, they are made of microtubules of these, so these are proteins, and this region, centrosome region with these centrioles, it is called the microtubule organizing center, which means from this region microtubules can arise, and they result in the formation of spindle fibers. So we will be talking about the cell division shortly, in which you will see how these spindle fibers are formed, and together they make the mitotic spindle, and this mitotic spindle helps in the separation of the chromosomes. Then last one is cilia and flagella. Now cilia and flagella, these are appendages on the surface of the cells. Like flagella is the long appendage, cilia are the short appendages, and they are all over the cell of the organism, and they help in the movement. So these were your non-membranous organelles in the cell. Now let's talk about what is inside the nucleus. Now nucleus is a very important organelle because it's a control center, because the DNA is there in the nucleus. So this nucleus is covered by a membrane, and it is called nuclear membrane. So you can see here, this is the nuclear membrane, and there are some pores in this membrane through which proteins and some other the mRNA can move out. So things can move in and out through these nuclear pores. Now what is this nucleolus? Nucleolus is here. This is a region in the nucleus which is responsible for forming rRNA. rRNA, ribosomal RNA, and this ribosomal RNA is one of the component of the ribosomes, and ribosome is a protein factory. What is nucleoplasm? So this jelly kind of a material here is called the nucleoplasm. Chromatin. Now you must know what is the difference between chromatin and the chromosomes. So now you can see that our cells are very small, and the nucleus is even smaller. So how does this long molecule of DNA, which is responsible, which makes up our genes, how this long molecule of DNA gets accommodated inside the small nucleus? So this DNA is basically wrapped on some proteins. So the DNA is basically, this long molecule of DNA is basically wrapped on the proteins. This is the way how long molecule of DNA gets accommodated inside the small nucleus. And when the cells are not dividing, this DNA wrapped on these proteins is like a dense mass, and it is called chromatin. But when the cells, they have to divide, then this chromatin has to get condensed, and it forms the chromosomes. So it takes the shape. So every species, they have a specific number of chromosomes. So these chromosomes are visible only when the cells are dividing. Okay, so chromatin gets condensed, and it takes the shape of chromosomes only when the cells they have to divide. We will be talking about more about chromatin and chromosomes when we are going to talk about how the cells divide. Okay, so let's now talk about the main differences between the plant cells and the animal cells. And both the plant cells and the animal cells, these are eukaryotic cells. Now plant cells, they have cell wall, they have chloroplast and the central vacuole. Now plant cells, they have cell wall, animal cells don't. So because of this reason, plant cells are very big and they have a shape. Animal cells are small and they are round. Chloroplast, plants have chloroplast, and this chloroplast has a special pigment called chlorophyll, and because of this pigment these plant cells can entrap the sun's light and they do the photosynthesis. The animal cells, like our cells, we don't have chloroplast, we don't have the chlorophyll, so we cannot make our own food. Plant cells, they have one big central vacuole in which it stores a lot of water, and the animal cells, they don't have the central vacuole. Now animal cells have centrioles in the centrosome region, but plants don't. So plant cells, they have centrosome region, but they don't have these centrioles. Animal cells have these centrioles, and animal cells also have lysosomes. Lysosomes, if you remember, they are the garbage disposal system, and plant cells, they don't. So let's see the differences again between plant cells and animal cells. Cell wall, yes, it's in the plant cells. Central vacuole, yes, it's in the plant cells. Chloroplast, yes, it is in the plant cells. Centrioles, it is in the animal cells. So this was all about the differences between your plant cells and the animal cells. So the last thing in this lecture is cell division. So first of all, cell division, which means we are going to talk about how cells divide. So let's see, we need to know why cells divide and how do the cells divide. Okay, so why do cells divide? Because okay, to understand why cells divide, you must have observed that like when we are born, we are a small baby, then we become an infant, toddler, we go through all the stages, and then we, at every stage, if you see, we are basically increasing in our size, we are becoming bigger and bigger. Now how is this possible? And also, if you have seen, like sometimes we get a cut or some wound, after some time that wound gets healed. How is that happening? It's happening because of cell division. Our cells are dividing. So our cells need to divide because we need to grow and develop. Sometimes we get wounds, those wounds needs to be healed, and lastly, sometimes some cells needs to be replaced. So all these are all reasons why our cells divide. Now how do the cells divide? Now there are two kinds of divisions going on in our body, mitosis and meiosis. Meiosis is a division by which our gametes are formed, and mitosis is a division by which our body cells they divide. So the basic thing what happens is, so we will be talking about mitosis, by which our body cells they divide. Body cells means your somatic cells, like your skin cells, the cells which track your digestive tract, the respiratory tract. These are all our body cells. So the main thing in mitosis is, first of all, the cell is going to duplicate everything, which means it is going to duplicate its DNA, it's going to duplicate its organelles. Once everything is duplicated, then it is going to divide them. That is what is the main logic behind the division, the mitosis. So everything, the cell is going to duplicate, and then it is going to divide it into two cells. So the division basically involves, first of all, the division of the nucleus and then the division of the cytoplasm. So this is the way cells they divide. Now let's see the stages of the life of the cell. The life of the cell basically means the different sequential events through which the cell passes. That is called the cell cycle. So basically the cell spends a lot of time in doing the preparation for the upcoming division. So cell spends 90% of the time when the cell is actually preparing for the upcoming division, when it has to form two cells from one cell. So it spends 90% of time in that. So it's in that 90% of the time, which is called the interphase, it is basically duplicating its DNA, it's duplicating its organelles, and then once everything is duplicated, then first of all it's going to divide its nucleus, which is mitosis, and then it is going to divide its cytoplasm, which is the cytokinesis. So in the interphase, basically you can divide the interphase into three phases. It's G1, S and G2. G1 is gap 1, S is synthesis phase, and G2 is the gap 2 phase. So basically in the gap 1 phase, cell is duplicating its organelles. So it's going to make, if it has one mitochondria, it's going to make two mitochondria. If it has one chloroplast, it's going to make two chloroplasts. It also duplicates its centrosome, which has those two centrioles, and you will see shortly those centrioles are responsible for forming the mitotic spindle, which is followed by your S phase, which is synthesis phase. In the synthesis phase, the cell is going to duplicate its DNA, and in the gap 2 phase, cell is going to make some proteins, and these proteins will be required for the upcoming division. So this is interphase, when the cell is preparing itself. Followed by interphase will be your cell division mitosis, in which involves 4 stages: prophase, metaphase, anaphase, and telophase. And this is mitosis, division of the nucleus, and it will be followed by the division of the cytoplasm, which is cytokinesis. And some of the cells, they don't divide and they go into the G0 phase, which means they're not dividing cells. The cells like nerve cells, the muscle cells, they don't divide, so they go into G0 phase. Now, when the cell in the interphase duplicates its DNA, the duplicated chromosome will look like this, but this chromosome will be visible only in the prophase. So otherwise, when the cell is not dividing, the DNA is in the form of chromatin, which is a dense mass. But when the cell is dividing, there is appearance of these kind of structures, which are called chromosomes. And the duplicated, once the cell has passed through interphase, when the cell has passed through the S phase, synthesis phase of the interphase, when it has duplicated its DNA, and in the prophase, when that chromatin gets condensed, it will take the shape of these. It will take the shape of this chromosome, and this duplicated chromosome will have two arms. Like this is one arm, this is the second arm. This is actually a copy of this. So whatever the gene is here, the same gene is here. Whatever gene is here, the same gene is here. So this chromatid and this chromatid, they are copy of each other, and when this copy was made, it was made in the S phase of the interphase, and these two chromatids are called sister chromatids. So this is the structure of a duplicated chromosome. So duplicated chromosome has two arms. One is this, another is this. This is a copy of this. When these copies were made, they were made in the synthesis phase of the interphase, and these two are called the sister chromatids. So now this is the cell cycle. You can see this is all interphase. So cell spends 90% of its time in the interphase and only 10% of the time in the division. Mitosis, the division of nucleus, and cytokinesis, the division of the cytoplasm. So now let's see the phases of the mitosis, the division of the nucleus. So interphase, you know that in the interphase, cell is preparing, it's duplicating its organelles, it's duplicating its DNA, and it's also it's making some proteins which will be required for the further upcoming division. So the mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. So remember that when the cell is in the prophase, it has already passed through interphase, which means the preparatory phase is over, it has prepared everything. So when the cell is in the prophase, there will be some events which take place. So this nuclear membrane will start getting fragmented, nucleolus region will disappear, and this chromatin, you can see this is all dense mass, it will start taking the shape, it will start getting condensed, and these are the centrioles, these are the centrosome regions having the centrioles. So as you know, in the interphase, they were also duplicated, they will start moving apart, and these are the microtubule organizing centers. So the microtubules can be formed from these centrioles, and they will start moving apart, and they will start forming the mitotic spindle. So because the spindle fibers can be formed from these centrioles. So this is the prophase. These are the important events which take place in the prophase. In the metaphase, this spindle apparatus is already formed, this mitotic spindle is already formed, and all these 46 chromosomes, they're duplicated and they come in the center. Okay, that is the most important thing which takes place in the metaphase. Metaphase is followed by anaphase. In the anaphase, basically there will be the separation of these sister chromatids. So you can see all the duplicated chromosomes, all the duplicated 46 chromosomes, they come in the center, and every duplicated chromosome has 2 chromatids. So one chromatid will go this side, another chromatid will go this side. For this chromosome also, one chromatid goes this side, one chromatid goes this side. So in anaphase there is this separation of the sister chromatids. So in the telophase, basically the 46 chromatids reach this side, 46 chromatids. So after anaphase is done, 46 chromatids reach this side, 46 chromatids reach this side. So every chromatid is going to represent a future chromosome. So 46 on this side, 46 on this side. So there will be formation of a nuclear envelope around these 46 chromatids, and there will be formation of 46. There will be formation of nuclear envelope or nuclear membrane around these 46 chromatids, and there will be the appearance of these nucleolus in this nucleus, as well as the appearance of nucleolus will be on this side also. And these spindle fibers, they will just disappear, and the chromosomes, they will start getting decondensed and they'll start becoming chromatin again. So so if you see a cell in telophase, it will give you an appearance of a very big cell with two nuclei, because nucleus has been divided but the cytoplasm has not divided yet. So basically the cytokinesis, which is the division of the cytoplasm, it actually initiates at the anaphase, and then it continues more in the telophase, and then after the telophase, this cytokinesis, it continues and it will keep on going till the two cells are formed. Okay, so cytokinesis initiates at anaphase and it continues even in telophase, and it continues even after the telophase, and it will keep on going till the two cells are formed. So this is the way two cells are formed, and this was the mitosis. So this is a division by which our body cells or the somatic cells, they divide. And the most important feature of mitosis is, from one cell two cells will be formed. They will be genetically identical to each other. Okay, so that finishes this lecture.